So hello everybody. Today we have a kind of informal meeting on the Kaggle competition, open problems, single cell perturbations. So the plan is to discuss something what we already have in public solutions, some ideas, some pains or whatever. Yeah. So maybe I'll start with some kind of slides which I a little bit prepared. Just a moment. It's like this. Oops. Okay, do you see my screen?
Yes.
Okay, so maybe just for the beginning, yeah, so just for initiate the discussion. So I don't know what pains, colleagues, do you have during this competition? For me, I think the most kind of two painful questions are the following, that I think there is no good correspondence between the cross-validation and the leaderboard score, and that creates, I think, certain problems, and I think eventually we'll have a shake-up at the end. So I think it would be quite impossible to not to have it. And another pain, I haven't I don't have some clear ideas how should we incorporate all this biological data which we have and which will be important for the judge prizes. Yes, in particular, so judge prizes include the questions about the attack-seq data, for example, and about the single-cell RNA sequencing data. So currently what we have is a pseudo-bulk data. We need to predict for the cell type, but we have the data on each particular single cell. How can we use it? I don't have some very clear ideas, and if somebody have one, it would be great to discuss. Maybe the other things, just what we can discuss, that there are some ideas, what we have in some public notebooks, how the top public notebook organized some of the cross-validation schemes from Ambros and MT. Antonina maybe can present her findings that the data is a little bit strange, that for some samples, cell types and drugs, we have originated from very few single cells. And probably these samples we should not trust very well. I hope Antonina will shed a small light on this point. And also we can discuss some further ideas from the notebooks and from the discussions on the forum. Okay, so we are very informal, so please kind of, if you have any comments, ideas or suggestions or questions, whatever, don't hesitate. Okay, so any questions? No. Okay, so if Antonina, can you, is it okay for you if you maybe start with your...
Just jump from the cells, right?
Yeah, okay.
Maybe I will show some recap about setup. Can I share screen?
Yeah, I'll stop sharing, and now we can share.
Disabled.
Sorry, just a moment.
Okay. So just a few words. I did get that some of you just new by here, so maybe don't know what happens at all. So a little recap for the setup of experiment, so you can understand the question about cell counts. Here we have plates. Each plate have several rows and a lot of wells, and they put cells into each well, so that each well contains almost all cell types. We have 6 here. And they also say in the description, the hosts, that there may be cases when drugs produce toxic effect, and there can be not all cell types, but some can be missing. So, for example, this well can contain three cell types. This one can contain 4 cell types, like so, and a lot of cells in each well.
Antonin, just a
all
question.
this,
So for
right?
these wells, it is something like 1 drug per well, or it's not 1 drug per well?
As you can see from this picture, is each well was treated with different drug, and 3 columns in this plate were treated with controls. They explain this so that there may be some effect of the well because of the way they process data further, but to avoid this unwanted effect, they treated entire column for each at least 1 well from each row. So many drugs and at least 1 control. Here we see this control is column. So we have drug and corresponding control.
Is it a positive control or
same.
negative control? Antonin.
Here 2 columns, 1 for positive control and 2 for positive. We have 2 controls and 1 negative control, which is just a solvent.
Okay.
The effects of solvents were just subtracted from the effects of drugs. This is a negative control. And positive, they needed positive control to be sure that there is some effect. So cell is alive, like so. Something happens here. Be sure of it. Yeah, and after this, not simple processing steps they had, and the data were then aggregated. So from a lot of cells, we have only just 1 number for cell type, like after all the steps for computation, aggregation, lemma model, and finally p-value. Anyway, we have 1 number for each cell type. But initially there were very lot of cells in each well, which were aggregated. And what I wanted to show, to discuss with others, actually.
Can I ask?
Yeah.
So it's after the treatment, the cells were sorted with the sorter, and then they read the RNA per cell, or they read RNA from the well, and then mapped it bioinformatically to the different types of cells. I don't understand how do they determine that this particular cell type has such a transcriptome.
I think, in my understanding, they collected row counts from each cell, and we have this data in a special additional file, data file, row counts per cell with cell ID. Yeah, and then cell data were aggregated.
So the cell type is
types
determined via their phenotype in the cell sorting technique, like cytometry or something.
I think
how they did annotated cell types. It's a big question, actually.
I think it was done computationally by the RNA sequencing. When you look on RNA sequencing single cell data, it's more or less, in the first approximation, it's easy to assign the cell types. Of course, it can be not very precise assignment, but I mean,
Okay.
just use a standard tool and you get something. And
So now,
Yeah,
I
based on
believe
gene
it's
expressions, like so.
it's sort
Sorry.
of encoding the data with the same thing which we want to decode. I mean, the genetic data of cell type and the genetic data of the change in phenotype of the drug is kind of I don't know, it's confusing.
Kishan comments that it's probably called cell multiplexing, that you can mix all the cells from different cell types, multiplex them, but then you can computationally distinguish this as B cell, T cell, because you can see it even on the clusters on UMAP. This is B cell, this is T cell. So these classical pictures.
Yeah. Yeah, they treated cells with a drug and with a solvent, and we have like some kind of difference between these two effects, and this is drug effect for the drug effect to expression, and then there were additional computations which I didn't want to discuss now because actually, but finally they got some confidence p-values which are just like our confidence on the presence or absence of the drug effect compared to its solvent, like this. Hope it's clear. Maybe not. Okay.
Yeah, I believe it's clear and understandable setup.
Nice. So the question here is when we look at the data which they collected from this experimental setup. So this a data ops meta file contains observational IDs cell by column. There are so many cells. I am bad with numbers, sorry. You can see. And library ID, plate name, you saw plate here, they have 6 plates. Well number, row number, column number in this plate, all about plate. Cell ID, we don't need it at all because all have PBMC blood cells. Donor ID, which can be important, they had 3 different donors, and for each donor, this setup was repeated with the same layouts and same conditions. And here we have cell type, some additional information we don't need it now, and the drug name. This, for example, this drug, we don't need any other of these. Actually, 24 hours, not control, dosage, don't need it. And SMILE's chemical structure of molecules. So, for example, this drug, they treated with this drug T cells. Actually, one this cell which was annotated as T cell DC4, it was from donor 2, it was from these coordinates in the plate, and you can see it, all this data. So totally you have 200s, right? 40, anyway, this number of cells, right? 6 different plates, 6 different cell types, and 147 drugs, and 3 of them are control drugs. And what I wanted to show, if we look how many rows, meaning cells, each drug has, we see there are very strange small numbers here, because this drug alvocidips was its effects were measured in 51 cells. That's very little amount because we have 6 cell types, and you can see this. So here I plotted number of cells per compound, and we can see 3 controls. High columns here, high bars here are controls. They treated with controls almost all of the cells, most cells. So we cannot see any differences here because 3 controls have much more cells and sampled. And if I
Daniel,
remove
if you have a
control,
question,
yeah,
just ask
I
the
have
question.
a brief
just
comment.
interrupts. Yeah.
Oh, excuse me. Okay, the alpha-CDIP is a CDK9 inhibitor which blocks transcription. So I don't think it's wondering that we don't see enough transcripts after the alpha-CDIP treatment in 1 day. So maybe I don't know if it's cytotoxic and kill the cells completely, or they just do not synthesize RNA. So we can await this result from this drug. And
I think it's not that much simple, Daniel, because they it depends on the dosage. And I'm not sure how they choose the dosage. I think they
this is an nanomolar inhibitor, so we should look, of course, at the dosage, and it blocks transcription, I believe, of a very enough strong to prevent the cell.
No, but I don't know. It would be great to ask the organizers, because I thought it would be natural to put such dosage that it will not kill the drugs, and so the dosage, I think, should be reasonable dosage should be uniform in drugs, because one drug is strong, another is weak, and if you put the same dosage for each drug, of course, some cells will die completely. Yeah, it's not what we want to have in this data.
Okay.
Yes.
Yeah, that's
so
it would be logical, to adjust the dosage. So we have similar effects. I don't know, from drugs. Like no one would kill a lot of cells, but we here see that some of them really kill. If it is about toxicity, which I'm not sure about, some of the drugs are really outliers here. They have really few observations. So most of the drugs have observations from around 1,000 of cells, right? And 4 of them very little cells. We can now break down more of these counts and see amount of cells per compound cell type pair. Right, we had pair compound cell type, right? And
Antonin,
here,
haven't you tried to exclude these drugs, which not so many cells, from the training set? Should it improve? Are these kind of outliers? Can we see that this data for these drugs?
I stuck here. I tried to remove 2 controls. We don't have DMCO, negative control, but I did try to remove positive controls, and I saw just worsening of my results, and locally and in the leaderboard. So I didn't try yet to exclude drugs with very little observations. I'm really afraid of excluding drugs because we already have very little data, right?
Yeah, but on that hand, if small data, it should create some kind of bad, some noise, and if you train on some noisy data. On the other hand, these drugs with many cells, why should we exclude them? Because pseudo bulk, when we make from many cells, it's very good. We should not exclude them, I think.
Yes,
I think the main
but
problem
they were. My idea was they were supposed to produce effect. It has to be different, and if we train our model with such outliers, which had supposed to produce effect with other drugs with unknown effect, there may be some bias towards these controls. I don't know, but it didn't work.
Okay, well, haven't you looked on some kind of, at least for this, our targets for these particular drugs, which have very small number of cells? Maybe we see something strange, something like the data are kind of outliers. So
no.
Can I
Not yet.
tell something about targets, and also, yeah, I should go in a short time, so excuse me for the interruption, and I'll briefly describe my idea. First of all, I saw the second outlier is a CDK1 inhibitor. It's again, I don't know why, maybe it's toxic too, and maybe this is because of the dosage of both these drugs. They really can kill the cells, and they're known to be toxic. And the second is I tried to describe the chemical space of the problem. And yes, we have a closed set of the regions, and we know that there will be no new compounds in the problem, which is on one hand, this is great because we don't need to mention the chemical space extinction and to manage drug similarity, and this is a bad because it makes the problem less interest for the computational chemists maybe. And first of all data are clear in terms of they have names one-to-one to smiles, so there are no ambiguous names or ambiguous smiles for different names, which are not very rare case. This is checked, and you can interchangeably use smiles and drug name and drug code in LIMS or something. Yeah, and the general idea, which I did not test, and I believe it can be helpful, is to describe not a general target provided by its developers, but the target described in binding DB database, which is collected, which contains more data about binding affinity of different compounds to different targets, because very much drugs, especially kinase inhibitors, which are the majority of the drugs, are known to have very wide affinity to some off targets, and the whole binding profile may be a better feature than the known target and the claimed target by its developers. So we can enrich the data set with the binding affinities of the particular drug to the known tested targets, and use the target distribution as an input for predicting the transcriptions.
Yeah, that's great idea, I think, but I don't understand. Actually, I don't fully understand how the features like this will looks like finally. So we have like we'll have
gene
like
name
data,
and IC50, for example.
Gene names, right? And
Yes.
0 and 1.
0, 1
by drugs?
or continuum. Or yeah, we can make a binary feature, active, non-active with some threshold. We can make a real feature in terms of IC50 or binding constant or something we can mine. First of all, I thought about using Kaggle, but Kaggle has a very bad API, and I don't really understand still how to get data from Kaggle and do not suffer, because yeah, they have very complicated queries to just take an IC50 from the experiment. And BindingDB has less data points, but they are clearer, and they can be easily obtained comparing to Kaggle. So yeah, I can
DB, right?
yeah, I can send you a link to this database, and yeah, and
It would be great. Yeah,
well, yeah,
I think we have to
honestly,
try
I
this.
honestly try to write the code, but unfortunately, I'm now still have no time to do it. Sorry.
Mm. I tried to get some info about drugs from drug bank, but I found only part of the drugs here. I'm afraid this will be the same for any other database. So we won't have full set of drugs. Anyway, should try with subset maybe, right?
Yeah. So that's
I see
all.
this
That
link.
also a couple of words about the chemical space itself. I did not see many interesting features in the descriptors or Tanimoto clustering or so, and I believe we cannot focus on this thing of the chemical data preparation, because from one hand we have narrow enough data set to ignore the Tanimoto similarity. I mean, in terms of we don't have very exotic species, and on the other hand, we have distinct set enough, not very tight clusters from structure point of view. So clustering, I think it's useless, but it should be tested of course, in real training, because I didn't try to use these features as inputs, but I just looked at the distributions and so on in my notebook. Yeah, so
Yeah, I also did try fingerprints from smiles, and Morgan fingerprints and Tanimoto distance was clustering very badly the drugs,
yeah,
so
that's the
it
point.
didn't work at all. But was interesting anyway.
Yeah, that's
Thank you
a
very
great
much.
point.
Yeah.
A great point from Kishan, that lipophilicity and metabolic reactions may be a good feature, especially I think lipophilicity, this because of its a direct metric of the cell permeability. But as we know, these are marketed drugs which may have not problems with bioavailability. So I don't think they will be differ severely in these terms. But yeah, I didn't test, so I just guess.
I tried to extract molecular descriptors which contain these descriptors for bioavailability and lipophilicity also, and I didn't get any good results with them as features, but I maybe did it wrong, because there were like around 167 descriptors, so maybe I did need to just filter or just select some most sensible ones. I don't know, because I had just made PCA for all and used as features, and the result was really bad, worse than zeros submitted. So
Yeah, they also have some correlated features, for example, solvent accessible area and polar accessible area and so on. But
yeah,
first of all, it's need to be cleaned. But the point, which from my experience, the 147 compounds is not enough set to feature can bear really helpful outcome. I mean, it's useful when you try to digest a large libraries, for example, 10,000 compounds. It's a first attempt to clusterize them to describe it. But if we have a perfectly described set of drugs which are marketed drugs, we don't yeah, we don't get some extra info from just descriptors. So
Daniel, just from your experience, for example, don't you see here something like two drugs which effect you would expect very, very similar? That we can test as a prediction. We can make just prediction for these two drugs almost the same, and to see whether the model improves or not.
yeah, they have, first of all, they have very close targets. CDK1 and CDK9 are the targets from one structural family, which is really narrow in terms of homology, and maybe also they
Sorry,
have
you say that who what drugs?
CGP 60474 and Alvacetic. The outliers which or
Okay.
kill either kill cells or block the transcription.
Mm.
Yeah, and also I can also look at the list tomorrow, for example, and describe the similar drugs. Yeah, I believe there are similar drugs here.
Okay. And another question: what do you think? How many genes we can expect affected by the drug? It can be 100,000
Directly.
or 10,000. What's the number you expect from your experience?
If we say about direct binding to the gene product, I mean, in protein, it's about tens, maybe some tens, up to 100.
I mean the kind of indirect, the effect which we see
Yeah,
here
okay.
on the transcriptome. So it's indirect can be, yes.
Yeah, it depends on the mechanism, of course, and if we block, for example, the cell cycle, it may be 1,000 genes. I mean, if we block transcription machinery, for example, CDK9 or CDK7, we can expect impact on all gene expression overall, especially if the gene is expressed intensively, because this drug perturb the transcription itself. So yeah, it's
for me it's strange. I thought maybe I'm wrong. I'm not that much expert. I thought that something like 100 drugs, 100 genes should be affected. But yeah, maybe what you say is kind of more correct, that all the genes can be affected. But
I'm not a biologist. Sorry, I just translate the things which our biologists say about such inhibitors. Maybe they will not be affected very strong, but there will be effect, especially if we block the signaling. I mean, anti-cancer drugs are usually focused on cell signaling, block some transcription factors to do their job, and each gene under the particular transcription factor, for example, TCF or NF kappa B, maybe also not a factor. Anyway, if we affect some signaling pathway, all genes under this pathway will be transcripted lower than usual.
okay,
But this is about in normal cells where they are usually upregulated. So how it be in blood cells, where some other pattern of transcription, I don't know how it would be.
Okay, thank you.
Thank you, everyone. I need to leave, and thank you for the discussion. It's very interesting.
Thank you for your comments. Very interesting.
Goodbye.
Bye, Daniel. But Antonin, you can continue. Yes, you have some. You're not.
Yes, actually, it's all about cell counts, and what just I just didn't expect this from such an experiments with competition. So we can see that some drugs, the effects of some drugs were measured from just one cell. There are several such drugs. So this one in T cells, only one cell. And this one in myeloid and B cells,
I don't quite
only
understand.
one.
So on the previous picture, I thought you say there are only 4 outliers.
Yeah, it's if we count how many cells for each drug,
Okay.
and independent each cell type, all cell types. But here I calculated considering cell types also. So
okay. Yeah.
we have pairs now.
Yeah. And so, okay, so you say that there are some pairs where only one cell. Can you show
one cell, right? Yeah.
Just a moment. Let me look on it. So we see some this MLM 2238, which we already see somewhere very nice stuff.
And here also, yeah.
Regulatory
B cells, two cells. That's all.
I think we should exclude them, at least try to exclude all the cells from the all these pairs from the training, because
I don't know. It's discussable because we don't have gene expression values. We have p values. So I think
it's
from two cell. Yeah. Sorry.
I mean, if you look on the single cell data, it's a kind of very dirty data with many dropouts.
Yeah.
But
and
then
two cells.
when you calculate the p-values comparing this single cell data for one cell for the pseudo bulk which is made from the 1,000 cells, this pseudo bulk doesn't have a dropout, and so you will compare something like one data which has zeros almost every random gene, and another which is quite good. Of course, many genes will be perturbated in this setup, but it will not correspond to kind of biological. No, that's my idea, but of course we need to check it.
Yeah, maybe I was going to exclude one by one actually, or maybe all ones or twos and etc. and see how it works.
All that less than 20, we should try to exclude, or maybe 10 or 5.
It's too much from we don't have anything after this.
No, I think it will be okay.
Yeah, you can see here's a summary table. So in T regulatory CD8 plus myeloid and B cells, all of them have drugs and treated in one cell, all of them. So only NK minimum amount of cell per drug is 2, and the best is for CD4. Maximal amount is also very different. Cells per compound, right? Per cell type. So it's
statistics. How many pairs have less than say 5 or 10, something like cells?
How many pairs? Didn't count it. Yeah, okay. Should add it. I will. Here I also have pictures, maybe a bit better than table. So breakdown by cell types is B and myeloid are our test. So little sample of drugs we have in train data, and from them we already seen this alvosically deep, right? MLN 2238, and this also. All were measured in one cell, and we have to like be kind of sure of effects. We have only 10%, right, for these two types, and 3 of them from 15. 3 from 15 were measured just in one cell, 1 or 2. It's really crazy for me.
Don't you have statistics for what is the average number of cells for the pair?
From this picture, I can figure like 150 will be
Okay.
made median, right?
Yeah,
So
so-
it's 200. So something like between 100 and 150 here, somewhere here, will be median. Mean, I did calculate it here. For myeloid cells, it was this number, 200 median cells per pair.
Mhm.
Okay, and same is for all 4 full cell types in train set. Maybe I can do it bigger. Like the first one is NK, and we just will see the y-axis here. We have around again 200 cells in all NK cell types, and in all CD4 we have even more, right? Around 400 cell per drug. If we look at CD8 plus, we see that the cell amount is really different from the 2 cell types above. It's around 50 cells, mean amount of cells, and we have outliers here in the other direction, so they were measured in much more cells than all other. So the data are very noisy. I don't know. And the last cell type, regulatory T cells, right? It's around maybe 30, 50 also cells per drug, and one very different IN 1451, and another one very different. We saw it maybe kill or produce toxic effect almost all myeloid cells, and in the other direction. So we have a lot of cell for this drug in T regulator. So maybe related somehow to the effects of these drugs, which is different toxic for myeloid and some kind of stimulating on T regulator. I don't know this biology behind this. It may be important, but no idea.
Yeah,
So
that's strange. Yeah, kind of.
strange. Yeah.
How can it be? So,
And yeah,
so there are some drugs which are many cells for one particular cell type and not for the other. Yeah.
yeah, and vice versa, right? Like one cell and all the other have very lot of cells. So if we think about this and go back to this picture, so we can imagine like in this well with this particular drug, they just collected one cell, and from the other well they collected 1,000 cells, and these data were aggregated. I don't know what to think about this. How to explain it? I don't know.
I think it should be effect of the drug. So I think they put in each well the same number of cells approximately, but some survive on the drug, but some not. But that's a little bit. It's a little bit strange.
Maybe we can. Maybe we can figure out some mean amount of cells they had at the beginning, and to add something to drug effect. So if there is very little cells, it's also an effect, right? And we can somehow consider it. I don't know how, actually. maybe I did try to add a feature composing of number of cells per compound cell type pair, and I didn't get good results here.
But we don't have this data for the submission part. How can we use this? can we use
It can be
definition?
calculated very easily. You just load this, as I show here. This a data ops meta CSV file, and you calculate number of rows for each pair, same name and cell type. It's 2 lines of code. Nothing
Just a
difficult
moment.
here.
So this file, you say it contains the information about the submission part also?
No. Only train pairs. I just used for test mean from train. So we have 15 drugs, which were tested in our test cells, myeloid and B, right? And we have number of cells for these pairs, and I took mean for all test. Yeah, it was mean.
Okay.
Yeah, so but the thing maybe is how to use this data. Maybe it will work somehow, but not the way I tried. I tried to add 2 target encoded features. I took 20 components for drugs and for cell type, and added 1 more column with cell counts. It worsened the results. So without these cell counts, I get better. Anyway, maybe somehow, maybe you can use somehow different this information. I don't know.
Do we know initial amount of cells in every well?
As I understand, no, we don't know.
So if we know it, we can maybe induce some survival rate variable, like relative amount of cells which survive the experiment.
Yeah,
but if we don't know,
sounds interesting.
So
we can make educated guess. But I did ask in the discussion somewhere. Yeah, in the discussion, I did ask this from the organizer. Maybe if you ping, I don't know how to tag hosts here, they maybe will answer. But here I did ask, is there any explanation of such a discrepancy in the number of cells of different types from which the data were collected for the drugs? So maybe we can assume that initial number of cells was maximal, right? So maximally you have like 20,000 with something around 20,000 of cells. It's a maximal amount for controls. Maybe this was initial amount, but maybe not. I don't know. And in some cell types here, like almost, you can say that almost all drugs killed almost all cells, right? Or why we see so little number of cells here?
Can it be very stupid that for this particular pair, they had several wells? No, we can check it with the because we have the well IDs for this.
Yeah, each well contained several cells, right? And what you
I
say?
think each well contains 1 drug. Yes.
Mm-hmm. Yeah.
And maybe for some drugs there were several wells. Just I don't know. Just for some.
For some drugs, several wells. I
Look,
don't think
for example,
so.
there are some colors which looks very similar. That's maybe that they are the same drug. One drug for two cells, full wells. I don't know.
I don't think so. I don't think so, because 72 per plate, it looks like just
Okay,
one well
I see.
for one drug.
Yeah,
We
okay.
can calculate.
Yeah, okay, I see. Yeah.
So if it's not toxicity, it's really a big question why such a discrepancy in amount of cells. But
And
if
another
it
question,
is,
and then
yeah.
we maybe can check donors. If maybe one donor have some mutation which makes his cells more kind of resistant to the drug.
Vulnerable to drugs, right? Yeah. Maybe,
So,
yeah, we should check it. Yeah.
So if this
Yeah, maybe.
peaks correspond to something like we see in one donor, it might be something. But otherwise, yeah, it's strange. Maybe we should ask the biologist.
We can see here that, for example, for this MLN 2238, here we have just two cells of B type, and each one was taken from different donor. So we don't have any data from the third donor. And here we have also 0 donor, but don't have third donor 2, right? Maybe you're right. Maybe donor 2 was somehow mutated, so his cells were all dead. Interesting.
Yeah, but it can be also random. No, I thought how to explain when we have this high number of the cells. So I think this is more probably related to toxicity, but
yeah, but this may be initial amount. So
but why it for some
like here it may be here we can maybe here we see initial amount of cells, so this and this drug did not affect any cell, so all alive
to some particular
all
cell
the
type.
other
Antonin, to some particular cell type, yes, you say.
For CD4.
Yeah, and for other cell type, this drug doesn't have this.
Yeah, the order, I guess,
that's
I have similar order. Yeah, no, if maybe, yeah, like here somewhere.
It's strange, I think.
Yeah, it's strange.
It would be also interesting to look at these peaks and split it by the donor. So is it split uniformly? So if we, for example, see that in this peak, only 2 donors, then it can be something related to donor. If it's not, it's approximately for the same number for all donors, then it can be related to the cell type, yeah. Because otherwise, no, that peaks is quite strange. So if there is a small number, it's easy to imagine that is a high toxicity, yes, for some drug.
Yeah.
But how to explain this big number, I don't understand.
Yeah. Yeah, I will add picture for donors, right? Good idea. Thank you. Yeah, maybe somebody have questions about it, some ideas.
Any questions, comments, suggestions, whatever? Alina, any remarks, whatever?
Maybe yes, we should get more information about initial amount of cells from organizers. Maybe it can be useful. And this number we can use to build additional features.
Yeah, it would be nice, but kind of I don't think organizers will share with us the number in the test, because
Yeah, will not.
that would be a little bit kind of how to say, we will get some information about the test we should not have.
Data leakage, yeah.
Yeah, some data leakage, yeah. Then yeah, but about the train part, yeah, why not? Why not?
It
And,
could be
do they
helpful,
get-
yeah.
On the other hand, I
Um,
think these experiments are quite standard, so we can ask any biologist who is, so these wells, I think they're quite standard by this 10x technology. So I think we can just ask any biologist who is working with this technology and just approximately how many cells they put there. Of course, it depends, maybe one, but if it's kind of standard in the field, then it should be known.
Yeah, this from the biological perspective would be anyway really interesting. Why some drugs, if it is related to drugs effects, right? Why they affect one cell type and that do not affect other cell type, or like here some drugs killed only three this cell type, but not this one. Yeah, very interesting. Maybe we can use it somehow, yeah. But if you don't have any idea, maybe we can discuss other stuff from your first slide.
Okay, so
What is it?
I don't know if people are tired, then maybe we can postpone it to the next discussion. Or...
Actually, I started analysis with CRAT for it's our package for differential expression analysis, and I used A data. So the data about raw counts after drug treatments, right? And I just plotted some pictures here, a little, but maybe already something here. Only I guess some % of cells, because it will process very slowly if I took all cells. It's very long time, so I just took maybe 10% of cells and plotted this picture, and features is number of genes, and counts is number of molecules, number of gene copies in each cell. So the distributions were skewed, right? There are few with very high amount of gene copies, and here we have subset with very large amount of genes in cell. Points, dots are cells. One dot, one cell. So it's also biased here. We see this bias, and we have I should train with numbers. So 426 to 5,000 genes per one cell. So some cell, in some cells, they measured, they were just 400 genes, and in other cells, there were like 5,000 genes. And from 1,500 to 28,000 gene copies, so counts itself. Very, very diverse, I would say.
But I think it's quite, for all single cell data, it's like this. If you look on some even tutorials.
Maybe, yeah. The previous competition, open problems, the plots were less skewed from this is different, actually, more noisy.
Yeah.
Than from the previous competition.
Yeah.
And also I have here number of features versus number of counts. So the more genes in the cell, the more gene copies. Like this, very, very high correlation. 96, 0.96 correlation between number of genes and number of gene copies. Yeah. I will publish this notebook soon. It is not finished yet. Maybe you can see something interesting here. Also, I calculated percent of largest gene, and we can see our familiar MLAT1, this nuclear gene, right? And B2M also from the previous competition, we saw them. And this RP ribosomal genes are prevalent also. So in most prevalent genes in the cell here. Most prevalent was MLAT1, B2M, yeah. So these statistics, I don't know how to use it. Didn't finish it yet. Maybe I will figure out something later.
Yeah, great. Okay, thank you very much.
It's like 2 of us here.
If you want, for example, I can share briefly go through my slides. If I don't know if you are not tired.
Yeah. I have.
Okay.
I will stop sharing, right?
Yeah.
Can I ask one question? Antonina, you have mentioned that we have only p-values, not counts. It's about what kind of data was?
We have to predict like transform p-values. So this competition
okay,
is is about prediction of our confidence in drug effects, like so. But also we have raw counts, but only for trained data. Raw counts after drug treatments.
Okay, I got it. Thank you.
Yeah. So I had this slide from the previous talk. So what's the p-values? They correspond to how much the gene is changing under the drug comparing to control. So you have something like untreated and you have treated, and you look how the gene is changing, and you calculate the p-value for this change. And there are many discussions, and as organizers say, there is no consensus in the community how to calculate the p-value. So they chose on one of the ways which seems most reasonable, but they also remark that kind of there can be lots of discussions about that, and on the video provided by the organizers, there was much discussion. But kind of
actually, if we just back to this one, I wonder how exactly we could recreate all the from the beginning. They say in this you have in this picture
they say
first
they share the
from
code.
hosts.
They share the
code. Share code, but we cannot use it because we don't have counts and any metadata about cells in test subset. So we could, but
no,
we can't.
We cannot. Yes, if we have test data, then there will be no competition because we can predict it. But we can try to reproduce it for the train part. Yes, but it would be quite hard. Yeah, all this reproduction with this limit.
I don't get it. So you take raw counts and data per cell, and what you predict from this?
You take a pair.
You can just
control and the treated by the drug for the same cell type. Yes, you have raw counts for these two things. Yeah, then you kind of try to look how it's changing. Each gene is changing. Yeah.
So I can reproduce the DE trained data,
Yes, yes.
but
In
how I can
theory,
use it?
but I think it's very complicated if you don't have experience doing this.
Why should I do it? They just said that we can go other way than predicting p-values. We can predict row counts for test, but we cannot because we don't have metadata for test cells. I mean,
Yes, yes. I don't know.
yeah.
Yeah, no, kind of. If we
So
kind of,
we just could.
if we can. So the problem is we do not have a correspondence, the cell before treatment and the cell, single cell, and after the treatment, yes? If we kind of computationally can say something like that this cell seems to be corresponding to this cell, at least, no, make some computational what looks similar, then you can do this, you can just train your model on single cells and try to, but I think it sounds a little bit crazy. I don't know how to.
Yeah. Maybe too much complicated.
Yeah, it's yeah. I don't have. Yeah, it's one of the questions. I don't have a clear idea how to use all this biological data. I think that's
Also, they have links.
Links, yes. Links. What's about links? There are 2 points. No, first point, it's a different technology made by different people, and I don't know, 5 or 6 years ago, and this changed everything because kind of, even if we want 2 different persons do the same, they get the different results in biology. But even more technically, so links is for cancer cell lines, and these are normal cells. Another reason links is for about 1,000 genes only. So here we have 20,000 genes, almost. Links is about 1,000. That's their point. They kind of say they can cheaply, very low expenses, measure these 1,000 genes. For about 1 million of small molecules. So links is great that we have lots of molecules there, but it has different cell types and different and only 1,000 genes. But links, of course, is very interesting to use.
Yeah. And we don't have p-values, right?
Yeah, exactly.
Only counts.
Yes, yes.
Yeah.
I'm not sure what we have, counts or something else, because it's not single cell technology, links.
It's not single cell at all.
Yeah, it's not single cell sequencing. That's as far as I understand, they have a little bit different in their protocol. That's why they can measure millions of the molecules. By single cell, it's impossible. It would be very, very expensive. But links is a special technology which you can kind of multiplex a lot. But yeah, but some people say that links data is kind of too much dirty and something like that. But I think it's interesting to try. Also, for example, for some drugs, like Verinostat, there are some even data sets on the Kaggle. I have put them for the single cell before treatment and after treatment, but for the cancer cell lines, so different cell types. And also kind of a little bit difference in technology.
So anyway, to imply somehow this data to enhance the trained data set, we need calculate p-values, right? Aggregated p-values.
something,
anyway.
yes. If you want to very precisely, yes. But if you want just on the level of something like, for example, it might be that we can see that if the gene is not changing under some drug for all this data, we can say that for our data, it should not also change, just predict as a constant. Don't bother about
Just
it.
как post correction, right?
Yeah. So
After
it's
model.
kind of a priori knowledge. If we see that kind of all the data sets says that, for example, this drug not changing the gene CDK, I don't know.
Okay.
Probably it should also work, should be true for our data, but yeah, but kind of we need to check this, all these things. Yeah. So
yeah, it's actually maybe a good idea. Also didn't get time to try to separate somehow genes, and this one I wanted to imply this differential expression analysis I do now for like selection of gene groups and predict genes by groups. So it may be one group like as you said divided according to the effect from other data sources, right? And there are many other ways how to divide genes for just most variable expressed genes or maybe
but
genes by enrichment.
Yes, but how, even if you have a group of the genes, I don't know, cell cycle genes, for example, so how it will help you? I don't quite understand. How do you want to predict them?
I thought to construct different models for each group, because it may be easier for model to catch patterns if yeah.
But what can be the reason why this should work? Because these genes are very correlated. But on the other hand, I don't know, when we do the PCA, for example, it also catches the correlations. It combines all the genes in some component, which, well, yeah, we should try, but yeah, I'm not kind of very sure.
One of the possible approaches.
Yeah.
Yeah.
I thought that kind of the drugs changing not so many genes typically, maybe 1,000, yes, but we have 20,000. Then we can think that changes of all the other 19,000 is something like noise. And just we can make something like just predict them by 0 and only predict something like 100 genes full. I thought, and to choose this 100, we can use some a priori knowledge, yes, some biological knowledge. But I haven't yet tried and not sure that kind of
actually
I don't see the reason why it should be better than PCA, for example, because kind of no PCA also in some sense does the denoising, and it can be seen by the model itself. Yes, some simple things in the data any model will see. We don't need to introduce a priori knowledge to see it, to help it. But okay, it might help. I don't know. Just an idea.
We have 1 more month to go.
Yeah,
right.
1.5, I think, or something. Yeah.
Right.
Okay. Maybe I don't know. Do you know this? Yeah, I think you know this trick. Yes. Just if you add this kind of priors, then the score is improved. Yes.
Sorry.
No, I think you know this. Yeah, that if you take your model submission, for example, do you see my mouse?
It's blending, with mean by cell and mean by drug, right? Yeah, I see.
So
I did try. Yeah, it improves score stably.
yeah. So thanks to Ludo
So-
Childiva, who is here on the meeting for this trick also.
Yeah.
And so the top public, as far as I understand, is organized something like that, that there is a neural network solution by Kishan, who was here. And then
Kishan was here, yeah.
you can do several tricks. At first, if you take this neural network and just calculate it with different random seeds many times, and then average, and you'll get the improvement of the score. And then you can
I tried to tune this one, this exact neural network, and I've got like very different results from 0.608 was the last one 608, and the maximal was 685, I suppose. Yeah, from same model, same features, anything the
neural
same. I
networks are
just
very unstable.
very
Yeah.
unstable network, actually. Yeah. Maybe I did it so unstable. I don't know.
I don't know. Yeah. On the
I
previous-
changed the learning rate. I changed architecture as he suggested. So blocked some layers or added new layers, and also I tried what else? Batch size. And I guess I tried to add regularization, L2 regularization in here, and nothing did work. Nothing. Nothing did it even so good as he did. 604, right? I got the minimal 608.
I think somebody got, I think, 599 something with this. Just when you
599. Yeah, I was not so lucky.
yeah, it depends on luck. Yeah.
Also, if you shuffle data with other seed, the result is also completely different.
Yeah. It's a kind of
My neural network is more stable, actually, but it's not public. Sorry.
Congratulations. Okay, I think maybe for today it's lots of information, so maybe we can discuss this later. I don't know, just maybe some examples. Yeah, so I compared the cross-validation scheme proposed by Ambrose and by MT, and here, and this is random cross-validation. And this, I changed the number of components for the SVD. And all this have that the best score is somewhere here. That is crazy. You need to took something like only two components. Yeah. We are typically taking something like 30 or 50, I don't know. But according to cross-validation, you should take something like two components for all of these cross-validations. And
actually, I often saw, like this one I saw a lot of times, and it never worked in leaderboard.
Yeah, so
See,
if you
show
submit
one way,
you get a bad leaderboard score. So that's kind of indicates that the problem. All the cross-validation scheme up to now, they have some problem. Yes, so we cannot completely rely on them. On the other hand, the good news that, okay, if you look on all, so the actual good point according to leaderboard, something like here, maybe 30 plus minus, doesn't matter. And we can see it more or less by all of the cross-validation schemes. They give different points. So for random, you should choose something like 23, maybe for this 35, but at least it's not the order of magnitude difference. And this will be good.
It may also depend on the model.
Yeah.
Because with your model, it may be 20, and with mine, it could be 50 or
sure.
more.
Just for this example, just one model, but only we change the cross-validation scheme, and we see that bad news that all of them give 2 components as a maximum. Yeah, and on the leaderboard it will be terrible. But
Mhm.
the good news that if you look on any of them, then all of them will say something like 20, 30, something like that. Not an order of magnitude difference. Yeah.
Yeah.
And the change, I mean, 20 or 30 or 50, something like, for this particular model on the leaderboard will not be great. It's not something like from 0 to hero. Okay, maybe I have more slides, but I think maybe we can discuss them at the next meeting, something like that.
Next time, right?
Yeah,
It's only
so
4 of us.
thank you very much for you and for everybody who participated.
Thank you
So
too.
maybe we can.
Maybe somebody can make another topic about cell counts, initial cell counts, because I did ask twice and nobody answered me.